It's true. Alright, whenever you're ready.
Alright, so for our project we made UV light. And UV light background, it's an electromagnetic radiation with a wavelength shorter than visible light. And UV light, the component of UVB is the harmful ray, and this causes sunburn and skin cancer. And these come from a tanning bed or the sun.
And how this causes a mutation in the DNA is it'll actually break the thymine bonds in DNA, and it'll basically, if it's like adjacent to each other like that, it'll break out from the one across from it, and then it'll like connect with itself almost, and it'll make a weird bulge in the DNA. So the DNA mutates and it doesn't divide correctly, reproduce itself.
And our hypothesis was excess exposure to UVB light over time will cause cancer because of direct damage to the DNA.
So for our experiment, we decided to do a cell culture, and for our materials, we used 20 petri dishes of one cell line of human skin cells, which means they're just from the same person. And then we used 3 UVB lamps and a microscope. And so what we did was we took 5 of the petri dishes of the human skin cells and put them under no light, which was our control. And then we put 5 under a lamp for 1 month, and then 5 under a lamp for 2 months, and then 5 under a lamp for 3 months. And then what we did was, after the specified period of time, we observed and measured the increase of division of the cells using a microscope. And then after finding that number, we took the mean of those 5 numbers and compared the means, and we used a
Two-sample t-test to see if it was a significant difference.
yeah.
Alright, so what we found from this experiment was that the more time that the dishes were under the light, it made the mitotic rate increase, as you can see from the mean.
Okay, explain mitotic rate before you...
It's how fast the cells divide.
It's like per 1,000 cells, how many cells are actually going under mitosis after a certain amount of time.
So the more time that the cell dishes were under the light, the more, like, the faster the mitotic rate increased. As you can see from the mean, it went from 50 to 154 by 3 months. And so what we concluded from that was that because the mitotic rate increased as the time of UVB exposure increased, we confirmed that the hypothesis that the UVB is a carcinogen because it causes growth increasing mutations in the DNA. And if the mitotic rate increases more than the control, then the DNA has an increased chance of cancerous mutations.
So the follow-up experiment was to take it from a cell culture experiment to an actual live specimen, to mice, and we basically do the same thing under just in a controlled environment, and the mice in boxes, and same controlled living style, except each box would have a different amount of time under a UVB lamp, and we would test whether or not they would get cancer. The previous experiment, we just saw the mitotic rate increase. It doesn't mean they actually got cancer, so we actually have to see if the increased mitotic rate will actually cause cancer in a living organism. That's it.
Okay, you guys have questions?
No, escape.
Jake's question.
So you didn't talk about this and it might not have occurred, but you said that UVB was the harmful UVBs, right?
Yes,
Yeah,
there can
so-
also be UVC, but that's less common.
And
Is it like UVA?
Yeah, UVA is the harmless counterpart to UVB.
Okay. Does it do anything to the skin or anything?
No, not significant damage.
Okay. Does it tan or anything?
ask Snooki. You can ask Snooki. That was our next follow-up experiment. Take the casters, you show her from a minute to any minute, see if that guy counts.
Okay, can you guys go back to your results for a second?
And we do see a slight dip in some of the trials, but because of, but the overall, the good thing is we did multiple trials, so we could see from the mean, on average, if the more exposure to UV light meant a higher mitotic rate. There could be there was some error in the experiment, but the trial numbers kind of took that part out.
Do you guys, oh, sorry, go ahead, Shayla.
Do you know if there's like a certain point in which the mitotic rate goes to a certain level in which you then begin to develop cancer?
That would be
Yeah.
a good
It's a
thing
good question.
to another experiment, but we do not know at this point.
So just so you are aware, I made this number up, this 50 per 1,000. I totally made that up out of the air. So that's not like a real number. It's just that the relative numbers are important for us to look at here. Does that make sense? I just made it up. Okay. I just want you to make sure that you understand that.
No, we
That's not
actually
like-
did the whole experiment.
No,
during
but
one
I
time.
mean that 50 per 1,000 might not be a normal, like the baseline. That might not be what really is the baseline. I just made that made that number up. Okay. So but what is good about this data is that you can see, and I think you guys drew a nice conclusion, that you do have a significant difference from your especially your 3-month
Yes.
trial compared to the mean or compared to the control, and I think that is a reasonable conclusion what you guys said about that. But I made the numbers up. Any other questions for these guys? Well done.